Hello, and welcome to the second lecture from Chapter 7. Again, we're talking about protein function, and in this talk, we're going to talk a lot about muscle contraction. And there are other ones. As you know, we talked about oxygen storage and transport, and in the next one, we'll talk more about the immune response. And again, here we're focusing really on the relationship of this diversity of structures and how they can give all these different proteins a diversity of function, and how the function both depends on the structure of a protein as well as the interaction that protein has with both proteins and other parts of the environment. So let's get into muscle. So first let's talk about the structure of muscle. So as you can see here on the left, we have just a whole muscle, and that's made of these bundles of muscle fibers that are called fascicles. And then the individual muscle fibers are shown over here. And I just want to point out, so no one is confused, that is calling a muscle cell and a muscle fiber are two words for the same thing in this case. So muscle cells are very, very long, as you can see from this picture, and because of that, they actually have multiple nuclei within one cell, just to make sure that one big long cell has enough energy. And we have these individual units for contraction that are called sarcomeres, which are shown here. And so, and the sarcomeres are what allow the sarcomere is the one individual unit for contraction within a muscle cell, and they sort of help to align that movement, so you have a lot of contraction that all happens at the same time. So all of your sarcomeres will start contracting, which makes the cell shorter, which helps to make the muscle shorter for contraction, and then they can also relax at the same time, causing a relaxation of the muscle as a whole. So let's talk a little bit about these myofibrils that contain sarcomeres as those functional units that we see here. So at the top we have a sort of real-life picture of what this might look like. And then we have this cartoon diagram on the bottom that goes into more detail about the different parts. And so over here in light purple, we have the thin filaments. And these thin filaments are all connected together at these Z discs. And then we also have these thick filaments, which are shown in dark purple. I'll just point it out over here because it's easier to label over here. But these are, all throughout the myofibril. And these thick filaments are made of a different protein called myosin, and they are connected together at the M disc. And so having these M discs and Z discs helps all of our thin filaments and thick filaments kind of act together at the same time to help pull the muscle all together within the one cell at the same time, within a sarcomere. So without knowing exactly how a muscle contracts, you might imagine that, oh, the way that the muscle contracts is by maybe having, a protein that's in a coil shape, and maybe that coil, like a spring, gets shorter and longer. But that's not actually what we happen when we look at the mechanics of muscle contraction. We actually see this sliding of the thick filaments among the thin filaments, and when they slide closer to one another, that causes an overall shortening of the sarcomere, and when they slide the opposite way, that can cause a elongation of the sarcomere during elongation, and that happens both due to nerve impulses that kind of get the process going, as well as this physical interaction of these proteins with each other in the thick and the thin filaments and the other components here that we see in the sarcomere. So let's talk about this sliding filament model in a little bit more detail. So at the top here, where we see number 1, we have the sort of elongated sarcomere, where the actin, the thin filaments, are sort of pulled apart relative to the thick filaments that connect between them, the myosin. And then you can see as the something is happening, right, that it's causing the thin and the thick filaments to sort of slide along one another and cause the sarcomeres to ultimately get shorter. And so ultimately we cause this whole process, the sliding filament model, where the thin and the thick filaments are sort of sliding past each other to make the sarcomere as a whole get longer or shorter. So let's talk more about what those filaments are made of. So like we just mentioned, the thick filaments are made of a protein called myosin. And here is a depiction of myosin, right? So it has these two sort of main components. We have this heavy chain, and we see 2 of them here. One's in kind of green and one's in this kind of light orange color. And they're fairly large in size. They're about 220 kilodaltons. And they have these sort of large, what we refer to as myosin heads, that when we see this sort of zoomed out diagram below, we see all these different pairs of myosin heads that are part of the heavy chains of the myosin. And the remainder of the myosin is mostly made of this alpha helix shape, where you have these two alpha helices that are wound around each other. The other main components of myosin are these light chains, and you can see we have 4 of them on one myosin. And each light chain is about 20 kilodaltons, so they're much smaller, hence light chains. And so when you look at a myosin molecule as a whole, we see on the bottom, right, we have all these different heads that are sticking out of this thick filament that we see down here. And so we have all of these alpha helices that are wound around each other, and we have these heads that are kind of sticking out from there. And then we'll see what the heads are doing in a moment. But before we get there, let's talk a little bit about the thin filaments. So the thin filaments are made of a different protein called actin. And actin is, as you can see in this picture on the right, has this mostly alpha helix sort of character, sort of similar to what we saw in myosin, with that same mostly kind of alpha helix character to it. But something interesting about actin is that it's a polymer, right? It's got stacks and stacks of these monomeric subunits that we see all in different colors here, and each one of them is sort of small and short. But when they're stacked on top of each other, we get these long, thin filaments. And so actin is used for a lot of the structural part of the cell, let's say. And a lot of times we're interested in building up or breaking down actin to change what a cell is doing, perhaps for movement, perhaps for other purposes. But in the case of muscle, we're actually looking at assembled polymerized actin filaments. They're not getting broken down or built up. They're just filaments of actin that are all ready to go. And the way we have movement of muscle is by the interaction between the thin filaments made of actin and the thick filaments made of myosin, like we have been discussing. So what are some other parts of the thin filament aside from actin, which we see as the backbone here in this drawing? We have another protein called tropomyosin. Tropomyosin is a protein that's mostly made of a coiled coil, and it's shown in the sort of bluish purple color here. That's tropomyosin. And it sits in the grooves of the actin filament, and it actually sort of hides the actin filament spaces from the myosin and stops that interaction of the myosin and the interaction of the myosin and the actin, which actually prevents muscles from contracting until they're able to be moved away or removed. And the way they're moved away is by another component called troponin. So troponin is a trimer, and it sits on the ends of the tropomyosin molecules on the junctions. And when they get a signal that's due to nerve impulses, a signal from a brain, they allow the tropomyosin to move off of that part of the actin filament and allows for the myosin heads to then bind to the actin, which ultimately allows the muscle to contract. So let's talk in a little bit more detail about that process of muscle contraction. So we have an illustration here of one sort of piece of the sarcomere. We have 2 thin filaments shown here. These are the actin, as we recall, and then we have a thick filament in between them, and that one is made of myosin. And so the way we can sort of think about the beginning of muscle contraction is when the thick and thin filaments are in this state, right? So you have the head of a myosin molecule, and it has this actin binding site where it's sort of loosely bound to actin, and it has this pocket in the middle that's available for the binding of a small molecule. And in this case, the small molecule that goes into that pocket is ATP. And so you may know that ATP is sort of used as the energy currency within a cell. And one reason for that is it has these 3 phosphate groups, and when these bonds are broken in between these phosphate groups, you get this big release of energy, which allows these chemical reactions to happen, all that good stuff. And so when we have ATP available, that goes into this binding pocket in the myosin head. And once ATP is then bound, the myosin head releases away from the thin filament. So we have this movement of the head down away from the thin filament. And so once that's ready, then we have this closure of the active site inside the myosin, and ATP gets hydrolyzed. So what that means is we have ATP bound to myosin in this top photo. In the bottom picture, we have ADP plus an inorganic phosphate. So we have the hydrolysis of a single phosphate group from that ATP. So it turns from a triphosphate into a diphosphate plus one inorganic phosphate group that's on its own, hanging out inside that pocket in the myosin head. And so when that happens, when the ATP gets hydrolyzed, the myosin head actually moves. It creates a different angle of itself in relation to the actin filament, and this movement is called the cocking of the myosin head. So once the myosin head is then in position, it is then going to weakly bind to the actin again. And recall here now we have this cleaved ATP, right? We have an ADP in here, plus this inorganic phosphate. It's bound weakly to actin. It's waiting for a signal to go. So when it gets this signal, we have a couple things that happen. So first we have this weak interaction of myosin and actin, and then we have this release of the inorganic phosphate, and once that inorganic phosphate is released, now we have this very strong binding of myosin to actin that we see up here, and it's very strong, but it's very transient. And once we have this strong interaction, we almost immediately have this movement back of the myosin head, and this movement of the myosin head is called a power stroke. And that movement is what causes that sliding filament action to happen, right? So the myosin, the thick filament, is moving in one direction, and it's pushing the thin filament in the other direction. So they're moving relative to each other, and ultimately this is what's causing muscle contraction. And so, and after that happens, after that power stroke, then we can have this release of ATP, and we're back to where we started, with this relatively weak interaction between the myosin head, which is sort of empty, it's unbound, and the thin filament above it. I just want to point out, a lot of times in the resting state, we're actually over here with this weak binding of ATP and ADP, and they're just waiting for that signal for the inorganic phosphate to be released, and to cause that power stroke. Although what we see here with the empty pocket is sort of the very, very beginning of the action, the way we described it. So how does this happen? Right? How do we get this massive change in the conformation of the myosin head that allows it to give us that power stroke that lets our muscles contract? So the way that happens is because of this amplification of small structural changes. So we have one conformation of this actin head in this pink color, the other conformation is in blue. You can imagine the rest of the myosin, the long alpha helices kind of extend past this way, and we're looking, in this picture overall, we're looking mostly at the myosin head. And in the middle here, we're looking at a bound ADP. And so when ADP is bound, and when you have the cleavage and release of that inorganic phosphate, you can have small changes in binding that sort of originate here. So you have this very small differences between the blue and the pink version of the myosin head that we see. But you can imagine a lot of these alpha helices, just sort of like we talked about when we were talking about in hemoglobin, a small change in one area can ultimately lead to larger changes in other areas as the alpha helices are sort of moving past one another, or as they might change from an attraction to a repulsion or a neutral interaction. And so over here we can see now this big change from the pink to the blue, which is what helps our myosin heads move so much during this process of hydrolyzing ATP and releasing that inorganic phosphate. So I know I just talked a lot about how this process works, but I think it's really helpful to kind of see how that is in motion. And so Dr. Middleton has picked out, and I agree that these are a really great resource to look at these few videos about muscle contraction. So let's go ahead and watch them together. So this first video really focuses on the signaling during muscle contraction.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger contraction. The muscle cells have an elaborate architecture that allows it to distribute calcium ions quickly throughout the muscle. Deep tubular invaginations in the plasma membrane, called TT, when the cell is stimulated, a wave of depolymerization, that is, an action potential, spreads from the synapse over the plasma membrane and via the tissue leads to the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent cytoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called the myofibril, the calcium interacts with protein filaments to trigger contraction. Each contractile unit, or sarcomere, stays active if thick myosin filaments are juxtaposed but cannot interact in the absence of calcium. This is because myosin binding sites of the active filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods in the cell, troponin oxygen moving from rod and non-myosin links. Opening the myosin membrane with idea, allows the membrane to swell and contract, resulting in its contraction of the membrane. Calcium is then quickly reflected from the reticulum by the interaction of calcium. Whether natural or non-hemoglobin, the membrane is well identified with tissue.
All right, so this video showed a pretty good sort of maybe further zoomed out level summary of contraction of muscles, and it showed us the importance of the signaling through nerves as well as via calcium into the actual muscle cells. That is what helps that muscle contraction cycle to get started. So let's now look at another video that really focuses more on that movement in the power stroke.
With calcium elevated around the myofibrils, myosin binding sites on the end are exposed, allowing interaction of the myosin cross-branches repeated, releasing the myosin cross-branch repeated with ATP. After ATP binds, it is split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to pump the cross-branch in preparation for the power stroke. The power stroke is initiated when the myosin cross-branch binds to actin. The cycle begins again when another ATP binds to myosin. At rest, most cross-bridges are in the closed configuration, propelled to the left of actin. The tropomyosin-troponin complex is moved away from myosin binding sites on actin by calcium ions.
So this was another really helpful short video that shows us the power stroke and the dynamics of what's happening with the ATP, ADP, inorganic phosphate, the resting position and that cock position, like we mentioned earlier. One thing I do want to mention about this video that is a small detail that is different from the lecture is that they don't show how that inorganic phosphate first gets released in order to initiate that power stroke. In the video, it shows the ADP and the inorganic phosphate releasing together, whereas those pictures from our textbook are a little bit more accurate that I talked about earlier, where first that inorganic phosphate is released, and then later at the end of the cycle is when the actual ADP is released. All right, so let's look at another video. This one is a little bit different than the others. So the others were sort of illustrations using cartoons and animation to sort of show us conceptually what's happening during our muscle contraction. This is actually primary data from a publication that is showing actual microscope imagery of myosin walking down an actin filament. So just to orient you, this long line here is the actin filament, and we'll see the sort of V shape on top of it, which is basically what the myosin heads look like at this resolution in this microscope. So let's go ahead and watch this one. And you can see in all of the different panels of this figure, you see this like V shape that's walking along the myosin, which is pretty cool that we can actually see this happening in real life. It's not just something we have to imagine. We know that this is exactly what is happening inside of our muscle cells when they're contracting. All right, so yeah, so then this brings us to I believe our final video, which is also more experimental data. And this data actually shows what's happening on a slide. So we have myosin, that's part of our thick filaments, that's tethered to a slide, that's attached to a slide. And then we have actin filaments that are not attached. And in this video, we'll be able to see the movement of those actin filaments along the slide due to its interaction with the myosin that's tethered and attached to the slide. So let's see what that looks like. All right, so yeah, again, some very cool, very interesting real life data from the lab. And again, I just want to reiterate that this is a little bit different from real life. We have myosin that's attached to slides, so it's not arranged in these very orderly sarcomeres and myofibrils like we saw in muscles. And that's what allows the actin to all move sort of together and linearly. Here it's just kind of myosin is randomly oriented when it's attached to the slide. And so we see this movement of actin that looks kind of random and all over the place. It's not orchestrated and directed in one direction the way you would see in an actual muscle cell. At any rate, I believe that was our last video, and so now you should be ready to answer the practice questions about muscle contraction.